And welcome to our next lesson, which is animal diversity. First, we're going to look at sponges, and then we're going to look at cnidarians. So sponges are, if you look at our cladogram here, they're multicellular and they have no true tissue. So they're the first organisms that we believe were developed. They're heterotrophic. That means they have to get their own food by eating other organisms, and they're from the phyla Porifera, and like I said, they have no true tissues. So sponges have asymmetrical appearance, meaning they can be all different kinds of shapes and sizes. It's the only organism that is considered asymmetrical, and it's composed of two layers of tissues of independent cells that have a jelly-like structure in the middle that holds everything together. It is important to note that the fact that it doesn't have any true tissues, it means that these organisms, the cells are kind of independent. They are not really working together. They're not, communicating and working as a team as much as everybody's just kind of doing their own job. So the two different types of cells that you will find is you have an outer layer of cells here, followed by an internal layer of cells here. Now, these internal layer of cells, which are found here, they're called collar cells. Collar cells are flagellated. That means they have a flagellum, and if you were to look at a picture of our ancient cells that animals come from, it would look very similar to collar cells. And in between this the outer layer of cells and the internal layer of cells, there are pores where the water filters through. The outer layer of cells' job is to protect the sponge, and the little flagella, the way that these cells work is, imagine that you're kicking in the water when you're swimming. So these work as little legs that kick the water that is filtering through the pores, causing a tremendous movement. So the water filters through in these pores. The little flagellated collar cells are kicking the water through and causes the water to come out of this central orifice called the ocelum. So this is considered the mouth and the anus because, as you can see, it only has one hole, and here's the central cavity where the water just filters through. It's very interesting as how this works as a filtration system. So because they filter like this, they're considered filter feeders. Filter feeders are organisms that are able to take massive quantities of water, travel through the pores, and then pick the plankton or small organisms that are found in that water, so that it can catch those organisms. And now we're going to watch a small video about how that works.
One of the most common sponges on coral reefs is the barrel sponge. Barrel sponges grow to epic proportions, getting larger than a person. Although sponges can't walk or swim, they can feed. They do it by filtering tiny plankton from the water. A sponge is covered with small pores called ostia, which lead to a system of internal canals and eventually out to one or more larger holes called oscula. Within the canals of the sponge, chambers are lined with specialized cells called choanocytes, or collar cells. The collar cells have a sticky funnel-shaped collar and a hair-like whip called a flagellum. The collar cells serve two purposes. First, they beat their flagella back and forth like fans to move water through the sponge. The water brings in nutrients and oxygen while it carries out waste and carbon dioxide. Second, the sticky collars of the collar cells pick up tiny bits of planktonic food brought in with the water. Sponges are very effective filter feeders since they're able to capture and eat particles as small as bacteria, as well as larger particles. They might not look like they're doing much, but a simple demonstration shows how effectively sponges can pump water. On a reef in the Caribbean, I make a dive with a syringe filled with a non-toxic dye called fluorescein. By squirting it around the base of some sponges, we can observe how the water is moving by watching what the dye does. Within only seconds, the dye is pumped through the sponges along with the water. As you can see, a sponge is a pretty good water pump and also a good strainer. Any plankton that goes in with the water won't come back out through the osculum. Tube sponges are even more spectacular to observe. They pump the dye so furiously that they look like a collection of miniature smokestacks. The ultimate test is a hefty barrel sponge. What will a big monster like this do? It takes a few seconds for the dye to work its way through the sponge. Wait for it. But then it pours out like smoke from a chimney. That's pretty good pumping from those tiny little collar cells.
So as you guys can see, the pumping that they do is very effective. So these organisms have also in between spicules and archaeocyte cells. So these cells are found in here. The spicules give, as you can see, these little shapes, they give structure to the organism. So it's another layer of kind of like a spiny material that gives them together, that holds them together. The archaeocytes actually are in charge of making those spicules as well as helping with the digestion and secretion of the material. So here is a better picture of what that jelly-like substance looks like, and these particles are actually, as you can see, they're like spread out throughout. So there's not a significant number of these cells that help the organism filter through. This is what the spicules look like. They're really tiny. This is under a microscope, and a network of spicules would look like this. So if you've ever found anything like this on the beach, it's actually the skeleton of the spicules. The last thing is the reproduction of the sponges. So the way that they reproduce is they have asexual reproduction where they can fragment, they can go through budding, or they can go through something called granules. Granules are like seed-like particles that form. Remember endospores, where something basically of the bacteria enclosed and kind of like the DNA enclosed in a very strong material so that the bacteria could come back when the conditions were better for that organism. So granules work like that. They're actually material or cells that are saved, and they form during challenging conditions so that then they can come back and reform the sponge. Sponges are also have something called generative properties, which means that you can tear them apart, you can put them in a blender, blend them up, and then let them sit, and all the cells that are left will come back and actually come back together and rejoin. So it's pretty cool. The sexual reproduction is interesting. So sperm is released into the environment, as you guys can see over here, and the sperm moves into another sponge, female sponge, that has the female cells, the eggs in here, and then those eggs get fertilized by the sperm that got released. So the fertilization actually occurs, even though the sperm is released, the fertilization actually occurs inside the cell. And then what happens is those larvae, which are already, fertilized eggs, leave the sponge and travel. This is the part where the organism is in movement when it's in a larval stage. So it swims for a few days. At some point it settles, and at this point it becomes what we call sessile. Sessile is when an organism does not move. So once it settles in the ocean floor, it will stay there and it will not travel anymore, and it settles in, and that's how the cycle works. So this is everything for sponges. We are going to go over